you can start.
Okay, thank you very much. First of all, I would like to say good morning, good afternoon, good evening, depending on where you are in the world for this symposium. And I would like to start my introduction speaking about my study species, which is the Mobula tarapacana, the sickle fin devil ray, which you can appreciate here in this picture. This is one of the largest species of mobulates that we can find, growing almost to 4 meters disc wide. And it's also a species that is characterized by its low productivity, meaning that it has very long pregnancy periods, followed by only breeding one very large pup, which is about like 110 centimeters disc width. And also then it has a very long resting period in between pregnancies, which could be between 2 and 3 years. This, along with a very large body, a slow swimming speed, a predictable habitat use, makes it a very easy target for fisheries, which is it's the main threat for this species. As such, the last report from the IUCN in 2019 classified the species as endangered globally. This species is usually solitary, but nevertheless it can form groups, especially in waters that are have high amount of nutrients. Like this species can be not only plantivorous but also ichthyophagous, and is primarily an oceanic species. Although, as I said, it can be sighted in productive waters, which can be also coastal waters or in oceanic archipelagos. This is a map from that IUCN report from 2019, where we can see the distribution of the species among all the oceans. And in orange, we can see the places where the sightings of these species are much more common. We're going to focus on the Azores archipelago, which is right in the middle of the North Atlantic Ocean. There is a seasonal aggregation in this archipelago every summer season every year. And the Azores archipelago is a very interesting spot, especially for marine megafauna. It's a biodiversity hotspot for marine megafauna. It's an ecotone that is literally in between the subtropical ecosystem of the Atlantic Ocean and the North Atlantic, functioning as a barrier and having a very high diversity for whales, seabirds, dolphins, sea turtles, and for sharks and large predatory fish, as we can see here, 80 species. One of the reasons why it's so rich in these species, especially the pelagic oceanic species, is because it has many seamounts distributed along the archipelago. We're going to focus mainly on the Princess Alice Bank, which is a seamount that is 40 nautical miles south of Fayal Island here. And this is the seamount where we find the aggregations of our target species, of our study species, the Mobula tarapacana. Here we have a very nice picture of this species with divers on the background. And it's basically this aggregation means, on terms of research, an incredible opportunity to gain knowledge regarding this oceanic creature, which otherwise would be very rare to sight just by going out into the open ocean. Here we cannot only see one individual, but dozens of them, and sometimes even hundreds of them at the same time, which is a very good opportunity to, for example, tag the animals with telemetrical methodology or use photo ID. As well, this aggregation at the Seamount, at the Princess Alice Seamount, suppose an incredibly great opportunity for the tourism, for the local economy. It yields a very great importance for the local economy here in Azores, dragging the attention of thousands of tourists all over the world that just want to come here to the archipelago and sight this wonderful species. As such, there is definitely a common interest in between science and tourism to protect and properly manage this place and this wonderful and incredible aggregation that occurs at the Princess Alice Seamount. To study the nature of this aggregation, we have developed a novel methodology, a new technology that uses basically an underwater camera that is located in the summit of the seamount and is recording all the water column all above until the surface. Here we have, for example, one of the frames of this, the images that the camera captures, where we can see divers, we can see a boat over here, and we can see our target species, the Mobula tarapacana. As such, this system has many advantages. At the beginning, for example, it's a non-invasive technology. This means that there is no interaction or whatsoever. There is no anthropogenic impact on the animals. And it's also a cost-effective methodology. This means that going into an offshore seamount, 40 nautical miles, from mainland, from the island, is very expensive in terms of gas, and it also allows you to have a very small window of time to sample in situ in the seamount. For example, if you take 4 hours to go there, then you have only 4 hours to sample, and then you need 4 hours to come back because you cannot be at nighttime at the seamount. But on the other hand, it also has its limitations. For example, this project has been running for years, but the consistency of the data gathering has not been always the best due to environmental conditions, due to failings in the deployments of the camera, etc. And also, we need to consider the subsampling. We're only sampling what we can see in the frame of the camera. We cannot see if there is a Mobula or if there is a diver or a boat outside the frame. And also the identification of other fauna, other species that we can see, because, for example, if they are very far away from the frame, from the camera, it's very difficult to identify between a tuna or between a seriola, an amberjack. I will continue to establish the objectives and aims of my study. First of all, of course, the aggregation of the Mobula tarapacana at Princess Alice. We want to understand the nature of this aggregation using an underwater camera. And we want to gain insight into the diel and seasonal pattern of abundance of this species in this location. As well, we want to analyze how the environmental effects variables, if they have an effect or not on this abundance, and as well the anthropogenic impact, the impact of the diving industry on this species. To begin with the material and methods, we go a bit further into the study site. As I already said, well, this is a map of the summit of the Seamount. We deploy the camera at 35 meters depth, and from there it records all of what it be the surface, the water column until the surface. Offshore Mobula system, OMS, that's how we call it, and it consists of the following. Let me start with the picture on the left. Here we have some batteries. These batteries will make the camera last between 2 and 3 weeks on the open ocean, and they are connected to this controller that we can see better on this picture. This controller basically tells the camera how to function. It produces the cycles of the camera. The camera has cycles that are 5 minutes on, 15 minutes off, and during those 5 minutes on, it's basically doing a time lapse, taking a picture every 5 seconds. This way, basically, we record during the whole day until the dusk or the dawn period, because the camera doesn't work at night because it has a sensor for the light. So when there is no light, the camera doesn't function, which makes sense because if it works at nighttime, we wouldn't see anything. Then we put the whole system with the batteries, the sensor, and the camera inside this housing that is fully sealed. And then further, we put it into this metallic orange tube to further protect it from anything that can happen in the ocean. As I said already, we deploy it at 30 meters depth. Basically, the system is around here, and it's attached to a mooring that is in the summit of the Seamount at 40 meters depth, more or less. And then it also has a rope that goes all the way up here to a buoy, so we can better visualize the system when we go after the 3 weeks or the 2 weeks and a half and we change the system. This buoy goes around to like 10 meters. And this is how the system looks in a bit more close look. This is the orange tube over here, and these are the divers, and this is how the camera would look at the surface. And we can see 2 screws, so the system to change is very easily. We just need to unscrew these things, and it's very buoyant. So it will come up, we need to grab it before it goes all the way to the surface, and then basically we just change it with the other system that we have. The data collection, as I said, has been running for years, but the 2020 data is the most consistent one. It's the only one as well that I need to remark that we have temperature and salinity data because we added a data logger into the system. And basically the variables that we have been collecting is the absence and presence of mobulas, the presence of divers, boats, other species, quantifying and identifying these species, etc. And the data analysis, basically, we are aiming to build 2 models. One model to explain the effect of the divers on the abundance of the mobulas, and one model to explain how the different environmental variables such as tide, temperature, salinity, or the different phases of the moon affect as well this abundance of our species. We will use these models with the 2020 data, and we will compare it to the 2018 and 19 data, which are less consistent. These are preliminary results over here, and I would like to commence with the patterns of abundance, particularly with the diel pattern of abundance of the species. On a first sight, like we cannot see a relative, like very well pattern of abundance during the day, but I would like you to pay close attention to the 10th, 12th, 14th, and 16th hour of the day, because these hours are at the same time when we have the divers going in and going out of the water. And we have a few peaks here, especially there is a diminish in the abundance of the species in between these 2 hours, which means that when the divers are in the water, not really close to the surface or going up and down the rope, the abundance decreases. Regarding the seasonal pattern of abundance, we can see that they are more abundant in August and less abundant in September. So along the season, the numbers are decreasing. We have a blank space here because the camera deployment failed at that time. And we can see that more or less the middle of the month of September, the Mobulas leave the Princess Alice Seamount. There is a preliminary result that is very important here for an anthropogenic model, and is how the diel pattern of abundance and the dive sessions correlate. And this is basically saying us something which is incredibly good for the touristic industry and is completely different to other studies that have been conducted before, and is that there is a probable positive correlation between the divers and the mobulas. So we hypothesize that the mobulas are so curious to the divers, to the bubbles maybe, that every time the divers are going down or are going up in the rope, the mobulas just come to greet them and maybe just be playful with them. That's also we hypothesize by the data gathered in situ, which basically is the best outcome possible for this study, for which is the diving industry. Also, there is there's application for other species in this study. Like we can gather so much data from other species as well, not only from the manta rays. Like for example, we have a lot of data from the season for the Seriola rivoliana, which is the amberjack, for another resident species in the Seamount, the Sphyraena viridensis, the barracudas. Then we also have data for very interesting ecological data for a benthic ray, which is inherent to the seafloor, but that has been many times sighted swimming in between the water column. This is Batistoidea centrora, which is like the long-tailed stingray. And also, I would like to mention, well, here we have a picture of another resident fish, which is the triggerfish, Balistes capriscus. And finally, I would like to remark how in the last year, in difference between the other previous 2 years, we have whale shark sightings. And a whale shark is very easy to sight. It's very easy to identify using this methodology. And just finally, to wrap up this presentation, I would like to come back again to how incredibly useful this new methodology is, how the invaluable information that we're gathering for this species, it's going to help us understand basically the aggregations, the nature of these aggregations, the patterns of abundance, and also, like, which is really important, especially for the local economy here in the Azores, which is how to properly manage this aggregation spot in terms of the diving industry, for which we see after this study is probably going to be a positive correlation in between them. Also, this methodology is very cost effective, so it can be applied for many different other species in different other places, for which we are very proud, and we should say that definitely this methodology has proven a suitability for studying these species in the Princess Alice Seamount here in the Azores archipelago. And with that, I would like to finish my presentation, and if you have any questions, I will be more than happy to answer them.